In this video, we're going to continue our talk on the cell cycle. Remember, we talked about the cell cycle of the animal cells, the prophase, the prometaphase, metaphase, anaphase, and telophase. Then in the last lecture, we forayed into the S phase, or synthesis phase of the cell cycle. And we talked about how DNA is synthesized during the S phase. Now we're going to come back to the cell cycle. And just as a reminder, the animal cells eventually become two daughter cells by what's called a cleavage furrow. The cleavage furrow is where contractile ring of microfilaments, right, actin and myosin, will cinch together like a tight belt and eventually give rise to two daughter cells. The cell cycle of plant cells, shown down here, is almost exactly the same as the animal cells. The only difference is when it comes to the final step of mitosis. Instead of a microfilament performing cytokinesis, there is a completely different mechanism of separating plant cells into two daughter cells. And this is the mechanism as shown down here. As you can see, right in the tail end of telophase, these vesicles that arrive from the Golgi apparatus start lining up in the middle of the two daughter cells, what's going to be eventually two daughter cells. And these vesicles start to fuse together. They eventually start fusing, becoming bigger and bigger, and at the end, what you're going to get is a completely new cell wall. And before it fuses, it's called a cell plate. So plant cells, cytokinesis was done by vesicles from the Golgi fusing together to form what's called a cell plate. And eventually forms a new cell wall. Remember, we talked about the cell wall containing a lot of carbohydrates. It's the vesicles that are coming from the Golgi. It's full of carbohydrates. And essentially depositing these carbohydrates in the space right here to create this new cell wall. Okay? So everything's the same except for cytokinesis. And here are some pictures of that, of the different phases: prophase, prometaphase, metaphase, anaphase, telophase. And you can see this cell plate starting to form during telophase. So we've been talking about the cell cycle of eukaryotes, right? We talked about animal cells 2 lectures ago, and I just mentioned the last step of the cell cycle of plant cells. But what about prokaryotes? Well, the prokaryotes undergo not mitosis, but what's called binary fission. And what happens in binary fission is that prokaryotes double in size and split in two. This is very reminiscent of mitochondria and chloroplasts, right? There are some eukaryotes that do binary fission as well. For example, amoeba. But there is a difference between binary fission between eukaryotes and prokaryotes. Eukaryotic binary fission, there's actually mitosis that needs to happen before the cell splits. Mitosis before. But the prokaryotic binary fission does not. No mitosis. What happens in the prokaryotic binary fission is illustrated on the left, right? Remember, we talked about DNA replication of circular E. coli chromosome. There's a single origin of replication, and here you can see the DNA is starting to peel apart and is going to copy and create 2 copies of DNA, circular DNAs. And somehow, in a mechanism that is still not well understood, the 2 origins of replication migrate to either end. So, origins of replication migrates to opposite ends. And finally, this pinches off to create 2 daughter cells. Again, the mechanism is not fully understood. Now we're going to switch gears a little bit and talk about how the cell cycle is regulated. And as you might imagine, the cell cycle is actually highly regulated. And it's important for several reasons. So the timing and the rate of cell cycle, how fast cells divide, are crucial to normal growth, as well as the development of the organism, as well as to maintain cells in the organism. Maintenance. And the big question is, what controls it? And I wouldn't fault you for saying, well, once the cell cycle goes, everything just falls in line and keeps going, right? That one cell cycle leads to another. But in the 1970s, people were starting to ask these questions, made the following observation. They took 2 cells, mammalian cells, one in the S phase, another one in the G1 phase, and artificially fused these cells together. What they ended up with was a cell with 2 nuclei. And when they fused a cell in the S phase to a cell in the G1 phase, the G1 nucleus was immediately forced into the S phase. So the G1 immediately went into the S phase. And in a similar experiment, they fused a cell in the M phase to the cell in G1 phase. And again, similar results with the M nucleus forced this G1 cell to immediately enter into M phase, with skipping the S phase. So there's only, as you can see in this cartoon, there's only one copy of the chromatid, but it still entered this mitosis phase, the M phase. These type of experiments led to this idea that there must be some sort of a cell cycle control system. So let's talk about the cell cycle control system. As shown here in the cartoon form. What you can see is that there are checkpoints. And these checkpoints along the cell cycle is where stop and go signals can regulate the cell cycle. And most cells, they have built-in stop signals. Most cells have built-in stop signals. That's because you want cells to naturally stop and only proceed if things are okay, if everything that needed to be done was done. Much like a car, right? In order for you to move a car, you need to step on the gas. If you let everything go, don't touch any pedals, the car will naturally slow down to a crawl. So you want the default pathway to be a stop and only proceed when you want it to. And this will become an important point when we talk about disruption of the cell cycle and development of cancer cells. And these go signals, go ahead signals, report whether or not everything is okay, whether crucial processes have been performed. And the most important checkpoint in most mammalian cells is the G1 checkpoint. It's the most important checkpoint in mammals. If cells in the cell cycle do not receive the go signal, the cells actually enter a G0 state. Remember, we talked about the G0 state. If no go ahead signal, then cell enters G0. And this is where the cells stop dividing. Cells stop dividing. And this is neurons, skeletal muscle. Those are permanent cells entering into G0. But some can be actually be called out of G0, like liver cells can be called out of G0. They can be in a quiescent state until some sort of signal gets them to start dividing again. And because this checkpoint is so crucial, it's called a restriction point. So what are these signals that are allowing cells to continue on their cell cycle? There are a group of proteins called cyclins and cyclin-dependent kinases that regulate this cell cycle. We're going to spend some time talking about this right now. There are proteins called cyclin that cyclically fluctuate in concentration, as seen here, right, in purple. You can see there's a peak of cyclin concentration. It's rising with the M phase. It peaks and then drops down. And then it starts rising again and drops down. And in conjunction with that, cyclin, there are proteins or enzymes called cyclin-dependent kinases, CDKs. Remember, ACE is a dead giveaway that it's an enzyme. Typically, all enzymes end with ACE. What kinases do is that they add phosphate groups. To proteins. And when you add phosphate groups to a protein, it's called phosphorylation. Or phosphorylate as a verb. Some proteins get activated when they're phosphorylated. Some gets turned off when they're phosphorylated. And as the name suggests, CDKs are cyclin dependent. It needs to bind cyclin to be active. So as you can see here, cyclin levels start rising at the beginning during the S phase, and it's peaking, and it peaks at M phase, and there's a sudden drop. Right? So this is the purple trace that we're looking at. And here, during the G2 phase, right, cyclin is now accumulating, and it's going to bind onto the CDKs and create what's called MPFs. MPF. It's called a maturation promoting factor. You can think of it as a mitosis promoting factor because the MPFs are required for cells to enter the M phase. What MPFs are is just cyclin plus CDK complex. So this MPFs now, cyclin and CDK, come together, and now they have, it's turned on its enzymatic activity, and it goes on and phosphorylates a variety of things. And one of the targets, it is the nuclear lamina. When phosphorylated, it starts to fragment, as in the pro-metaphase. And MPF activity disappears when cyclin starts to get degraded. MPF turned off by cyclin degradation. Then it goes back to just CDK without the cyclin. It's not active, and it waits around, sticks around until cyclin levels start peaking again, and this whole cycle can begin. And remember, I mentioned that this transition, this start of the G1 phase is so important. It's called the restriction point, right? The G1 checkpoint. It is so crucial that it has 3 different CDKs and several different cyclins controlling G1 phase, right? Exerting a tremendous amount of control on whether or not cells can enter the G1 phase. And once cells enter G1, usually it will go through the entire cell cycle. So this cyclin CDKs is one signal that's regulating cell cycle, but there are other signals that regulate cell cycle as well. And let's talk about some of the internal factors that regulate cell cycle. And one example that I want to bring up is that during anaphase. During anaphase, sister chromatids that lined up in the metaphase plane in metaphase begin to separate into 2 opposite poles. This is actually triggered by internal factors. It's not cyclin and CDK. I don't need you to know what they are exactly, but it's a complex of proteins that allow the sister chromatids to start separating and anaphase to proceed. And this complex of proteins, what they do, for one, it activates an enzyme called separase. What separase does is that it cleaves cohesins. Remember, cohesins are what keeps the sister chromatids stuck together, right? These are cohesins. So there's a special group of proteins that get turned on as a signal to say, let's proceed, which activates a separase, which comes and starts cleaving, that's my scissors, cleaving cohesins, separating out the 2 sister chromatids, and now they can start going to the opposite poles. So that's one internal factor that regulates cell cycles. There are also other external factors as well. And these external factors are called growth factors. And these were discovered in what's called a tissue culture setup, where you can grow cells in a petri dish. Cells do not divide if you don't give them enough nutrients, right? If you starve them of nutrients, they will not divide. But even if some of these cells are given full complement of nutrients, some of them don't even divide unless they're given this specific growth factors. What are growth factors? They are released by certain cells that stimulate other cells to divide. They're released by certain cells that stimulate other cells to divide. And there's over 50 growth factors identified. But the example that I want you to be familiar with is PDGF, which stands for platelet derived growth factor. So what is PDGF? PDGF, as the name suggests, is released by platelets. What are platelets? They are cells in blood. And what PDGF does is that it triggers cells called fibroblasts to divide. Triggers fibroblasts or fibroblast division. What are fibroblasts? They're connective tissue cell. You can isolate connective tissue from humans or from mice. You can cut them up, and then you can enzymatically treat them into single cells, and you can grow them in a tissue culture flask. But without PDGF, you give them all the nutrients that it needs, right? Amino acids, salts, sugars, whatever. But without PDGF, they do not grow. But as soon as you add PDGF to this media, fibroblasts, these connective tissue cells, will start dividing and growing rapidly in this flask. Okay, this is a situation in a tissue culture or in this isolated experimental setup, but what is it doing in real life in the organism? When there is a wound, you bleed, right? And platelets arrive. And they secrete or they release PDGF. And what PDGF does is to stimulate fibroblasts to divide. And what does a connective cell do? It helps heal the wound. Okay? So this is an external factor, right? PDGF is released by one cell type to influence the division of another cell type, fibroblasts in this example. Okay? So why are we talking about cell cycle regulation? Well, it's extremely important to know cell cycle regulation because during dysregulation, when cell cycle is not regulated, you get cancer. The hallmark of cancer cells is that they divide excessively, right? Sometimes kill. The organism. And cancer cells can divide indefinitely. What does that mean? Definitely, it becomes immortal. Usually, normal cells divide anywhere between 20 to 50 times before they die. But cancer cells, one of their hallmark characteristics is that they divide indefinitely. They never die. And the most famous cancer cell line is called HeLa cells, which was derived back in 1951. This is a cervical cancer cell line. Cervical cancer cells. And they were derived from a patient named Henrietta Lacks. And there's a fantastic book called The Immortal Life of Henrietta Lacks. I highly recommend you read this book. But we have the HeLa cells right here on Oneonta campus. Right? And this it was taken from Henrietta Lacks back in 1951, and the cell line is immortal. It keeps dividing and dividing and dividing. Many labs around the world use this human cancer cell line. So what happens when normal cells become cancerous? It's called transformation. It's a process where normal cells become cancer cells. They get transformed. And usually our immune cells are excellent at picking up these cancer cells and kill it as a foreign object. But some cells do escape this immune system. And they can either become tumor of one of two flavors, benign tumors, which stays put in the organs, versus malignant. Stays in tissue. Malignant ones spread throughout the body. And here in this figure, there's a tumor, breast cancer, which grows from a single cancer cell, and it keeps growing. And it's going to now, it's going to metastasize. Metastasize. means to move to another location. So it's going to leave the breast tissue through blood system or the lymph system and now find its way to another part of the body, grow, and spread as a malignant tumor all over the body. And breast cancer is extremely metastatic, extremely aggressive. It loves to go to the other locations. And if you don't catch it early, it can result in death. And chemotherapeutic drugs, chemotherapy, because cancer cells are cells that have lost its cell cycle regulation, chemotherapy targets cell cycle. An example of that is Taxol. Taxol prevents microtubule depolymerization. Remember, microtubules are required for sister chromatids to separate into the opposite poles, but it needs to eventually break down. And if you prevent them from breaking down, the cells will die. So it stops cells going to metaphase. Right? But the unfortunate side effect is the chemotherapy, not only does it affect cell cycle of cancer cells, it also attacks cells that are are healthy and dividing normally, right? That's why the side effect of chemotherapy is hair loss, memory loss, the immune system is going down. Every single system that requires new cells to be born, chemotherapy is going to affect, right? Because it doesn't know to attack a tumor cell versus a normal cell, right? That's why the side effects are so terrible. Anyways, so I think cancer sheds light on how important it is to know how cell cycle is regulated. And that brings us to the end of regulation of cell cycle.